The two stages are transcription and translation. Okay, transcription is the eukaryotic cells. Transcription occurs in the nucleus. Okay, DNA to RNA. It is a transfer of genetic instructions in DNA to mRNA. mRNA is a messenger RNA. Okay, where transcription is occurring. Okay, the next one is translation, RNA to protein, the process in which the genetic code in mRNA is read to make protein. Okay, those are the stages under protein synthesis. Okay, moving on to the diagram. Okay, you all can see the diagram of DNA replication and the diagram of protein synthesis. Okay, I'll explain you how these are the stages of DNA replication in diagrammatic form. Okay, so that you can have a clear knowledge of these steps. Okay, the first one is the DNA unzips. Okay, this double helix, it unzips. Okay, it breaks off. Okay, the 5' is the leading strand, and the 3' is the lagging strand. Okay, the leading strand, the DNA synthesizes continuously. Okay, it moves continuously, it does not stop. Okay, where the lagging strand, the DNA synthesized in short fragments, so-called Okazaki fragment. Okay, the Okazaki fragment is all this. Okay, it breaks off. Okay, when it forms, after that it breaks off, then it continues again. But in leading strand, it just continues directly. Right, it's continuously moving. Right, it does not stop. Okay, that's the meaning of leading and lagging strand. Okay, this is a clear diagram, the steps. It has 5 steps: 1, 2, 3, 4, and 5. The first one is the helicases. Helicases is one of the enzyme. Okay, helicases is one of the enzyme. Okay, the helicases, the work of the helicases, it unwinds the parental double helix. This is the double helix, and these are the nitrogenous bases. Okay, thymine, cytosine, and guanine, and adenine. Okay, these are the nitrogen bases, and this one is the helicases. Okay, it's present here. These two circles is the helicases. Okay, it unwinds this. Okay, it breaks off this. Okay, the second one is the single strand binding protein stabilize the unwound parent DNA. Okay, these binding proteins, it stabilizes, it protects this unzip double helix. Okay, this is the DNA polymerase. Okay, number 3 is the leading strand is synthesized continuously in the 5 to 3 direction by DNA polymerase. Okay, the leading strand. So this is the leading strand. Okay, it moves continuously, and this is the lagging strand. It forms short fragments. Okay, Okazaki fragment. Okay, number 4 is the lagging strand. It synthesizes discontinuously. Okay, the RNA primer, which is extended the DNA polymerase to form an Okazaki fragment. The function of this DNA polymerase is to extend the primer. This is the primer, RNA primer, right? The job of this DNA polymerase to extend this primer, okay, this RNA primer, so that it can form those short fragments, okay? And the 5th one is after the RNA primer is replaced by another DNA polymerase not shown, the DNA ligase join the Okazaki fragment to the growing strand. This is the DNA ligase. So the meaning of this last step is this DNA ligase, this RNA primer, or this DNA, or these short fragments, it will continuously moving by the help of DNA ligase. Okay, that's the steps. Okay, in diagrammatic form of DNA replication. Okay, the next one is protein synthesis. You can see. Okay, this is a cell. Okay, a cell membrane. Okay, in a cell there is a nucleus. Nucleus DNA is present, and the mRNA is present, and the cytoplasm, okay, is present all in the cell membrane. Okay, in the nucleus we know that DNA is present there because it's a genetic material. Okay, it's always present in the nucleus. Okay, by the work or by the work of this mRNA, transcription begins. This stage where it comes in one of the stage of protein synthesis. Okay, the mRNA forms, and after that it moves to the ribosome or the cytoplasm. After that, then the transfer RNA in translation occurs. Okay, where transfer RNA X or continues to work from there. Then after that, then it goes. It's a cycle. Okay, right, it's a cycle. In transcription, the DNA sequence of gene is transcribed into pre-mRNA based on the base pairing property of nucleic acid sequence. The pre-mRNA is modified to produce mature mRNA, which is then transported to the cytoplasm. Okay, those are the diagrams and the steps of DNA replication and protein synthesis. Okay, I hope students that you have understood what is being taught, okay, through this lesson. Okay, after this lesson notes and this explanation of the diagram, there's an activity, okay, to sum up this lesson. Okay, there are some activity, okay, that you students will do. Okay, it's a class activity. Okay, individual activity. Okay, here are the worksheets for you to do the activity. Okay, there are only 4 questions, okay, for you to answer. Okay, it's a 5 minutes activity. Okay, it's an individual activity. You'll do it by your own, okay, to answer those questions. It's a 5 minutes activity. You may start. You may start. It's only 4 questions. After doing those activity, then we'll be marking it together in class. Okay, 4 minutes left. Okay, 3 minutes left. Okay, the last 2 minutes left for you to complete those activities. Okay, students, have anyone finished those activities?
Yes, madam.
Okay, I hope that you have finished those activities. We can mark it together in class. Okay, the first question is to define DNA replication. Okay, if you have this answer, DNA replication is a biological process of producing 2 identical replicas of DNA. Then you can give 1 mark for that. The first question. Okay, the next one is to define protein synthesis. Okay, if you define that correctly, you may give another mark for that. Okay, another question is to explain the steps of DNA replication. There are 5 steps. Okay, if you have that correct, you may place a mark for it. Okay, it's half mark each. Half mark on those steps of DNA replication. Okay, the last question is to define the steps of protein synthesis. Okay, if you have that correct, it's 5 marks. The total mark is 5. Okay, you have to round the marks after marking. Okay, you may give the papers back to the owner. Okay, for them to see their marks. Okay, proceeding to our, this is lesson notes I've already explained you, and this a video, a short video for you to watch so that you can know more about this topic. Okay.
It might be hard to believe, but at the very start of your life, you're a single, microscopic cell called a zygote. Your body now contains millions of cells, which all came about by the process of cell division. Cell division happens when one cell divides to form two cells, and it is the basis of growth and repair in humans. It's also used for asexual reproduction in organisms like bacteria. But a single cell cannot just split itself in half to form two whole new cells. They would essentially only be half a cell. Before the cell divides, everything inside needs to be copied. This includes all of the parts of the cell, like the mitochondria, as well as the chromosomes inside the nucleus. A human body cell contains 23 pairs of chromosomes, 46 altogether. During cell division, the DNA in the chromosomes is copied or replicated to form 46 pairs, double the number of chromosomes. These pairs split apart when the cell divides to form two new daughter cells, each having the correct number of chromosomes. The splitting apart of the chromosome pairs happens during a stage in cell division called mitosis. Let's take a closer look at DNA replication. DNA is made up of many nucleotides, each containing a base represented by the letters A, T, C, and G. A piece of DNA contains two strands of nucleotides, twisted together to form a double helix. The strands are complementary. This means that whenever there is an A in one strand, it will be joined to a T in the opposite strand, and wherever there is a C, it will be joined to a G. If you are not familiar with the structure of DNA, then take a look at this video. During DNA replication, the double helix unwinds. Then, an enzyme called DNA helicase unzips the DNA, so the two strands are separated. This happens at several points along the DNA. An enzyme called DNA polymerase attaches itself to the DNA strands and is used to add complementary free nucleotides to the now exposed bases on both strands. A pairs with T and C pairs with G. This forms two DNA molecules, each of which have one brand new strand and one from the original DNA. These two strands twist to form a double helix. The two new DNA molecules are both identical to the original DNA molecule. In some ways, DNA replication is similar to transcription, a process that happens during protein synthesis. They both use enzymes to join free nucleotides together, and they both take place in the nucleus, but there are important differences. In DNA replication, each DNA strand is used as a template to make two new identical DNA molecules. In transcription, the coding strand of the DNA molecule is used to produce a single strand of RNA. So, to recap, in this video you have learned why DNA needs to be replicated before a cell divides and how this takes place.
If you liked the video, give it a thumbs up and don't forget to subscribe. Comment below if you have any questions.
I know that you already watched that video. Okay, I know that you understand more about this topic. Okay, because it simplifies or it explains more about this particular topic, DNA replication and protein synthesis. To sum up my lesson today, okay, by recapping, okay, what we have already learned, the DNA replication, what is the definition of DNA replication, protein synthesis, and the two stages, which is transcription and translation. The steps in DNA replication, there are 6 steps, okay, and plus the diagram that I've shown, okay. This diagram can help you understand more on DNA replication and protein synthesis. Okay, students, that's all for our class today. Okay, then you can go home and go back to the lesson that I've taught today, and you read again or read the textbook or you read your notes so that you can understand more on this topic, DNA replication and protein synthesis. Okay, that's the end of our class. Okay, you may set up your desks and chairs, okay, and wait, take out your books and wait for the next class. Vinaka.